Hello everyone. I'm very happy to present this video. In this video, I'm going to discuss applications of molecular docking studies in drug development. The use of in silico studies in drug discovery is very popular. Nearly every aspect of drug discovery is now explored using software first, and experimental work is planned based on in silico results. Although in recent years, we can see that more and more researchers are adopting molecular docking for drug development. However, awareness of molecular docking in formulation development remains quite limited. In this video, we will explore how molecular docking can be applied to research related to formulation development. Before I start with the discussion, let me clear that this video will benefit all those researchers who are involved in new product development. So whether you are from academia or you are from industry, these techniques are definitely going to help accelerate and enhance the impact of your findings. Now let's dive into the applications of molecular docking in drug development. But before that, let me briefly explain molecular docking technique to you. Molecular docking technique involves a study where the interaction between two molecules are studied. Typically, one molecule is small in size and is called ligand, and the other one is large in size and is normally protein. So we need 3D structures of both. The 3D structure of the protein can be obtained from the Protein Data Bank, which is a dedicated website for the 3D structures of proteins, and then this structure is prepared for docking using a specialized software. These software can be either Autodoc Vina, Discovery Studio, PyMol, Schrödinger or Molsoft. Similarly, the 3D structure of the desired ligand is obtained from databases such as PubChem, and the docking process is then performed. The docking results provide information on the docking score, which indicates strength of the affinity between ligand and polymer, along with the details about the nature of the interactions, such as whether the involved interactions are hydrogen bond interactions or hydrophobic interactions. To successfully perform this technique, you should learn it from experts or take advantage of online courses and YouTube tutorials. I have given link of some of the courses in the description of this video. We have explored what molecular docking is. Let's look at how this technique can be applied in drug delivery and formulation development. Often formulation strategies involve approaches like solid dispersions, complexations, or co-crystallization. This molecular docking technique can be used to predict whether a drug molecule and a selected polymer will interact favorably. Alternatively, if we already have achieved the desired results using a particular approach, docking can help elucidate the mechanism behind these interactions. Additionally, the technique can be used to predict the role or effectiveness of a stabilizing agent in enhancing the stability of a drug molecule. It can also assess the compatibility between a drug and excipient, and it can evaluate the taste masking effect of an agent. I will now explain these applications with an example. I am using a published research article for this explanation. This study is about enhanced solubility and dissolution of curcumin by a hydrophilic polymer solid dispersion and its in silico molecular modeling studies. The objective of this study was to enhance bioavailability of curcumin by preparing solid dispersions of curcumin with Udrugit EPO, which is a hydrophilic polymer. Essentially, in this article, they have prepared and characterized solid dispersions of curcumin with Udrugit EPO. Further, they evaluated these solid dispersions for their antioxidant and in vitro resolution profile. During the evaluation, they reported that solid dispersions of curcumin have higher dissolution rates and increased antioxidant activity. As mentioned clearly in the abstract, they have used molecular docking and molecular dynamic simulations to understand the molecular level interaction mechanism between curcumin and Udrugit. Let's see when and how they have done in silico studies in this project. First, they prepared solid dispersions of curcumin with Udrugit. The dispersions were prepared using two methods, roto-evaporation and spray drying. They also studied the effect of solvents on the nature of solid dispersion. They tried both organic solvents and water for this purpose. Then they used molecular modeling studies, means docking and molecular dynamic simulations to understand mechanism of binding between drug and polymer. That means they are using the in silico tools after they have demonstrated that there is an enhancement in solubility and dissolution. For the molecular modeling interaction studies, they used two techniques. One is molecular docking and other is molecular dynamics. We discussed briefly what is molecular docking in the beginning of this video. Now, how does molecular dynamic simulations differ from molecular docking? Normally in molecular docking, the polymer structure is kept rigid while the ligand structure is flexible. Why this is done? This is done to make the docking process faster. To put simply, we can say that the molecular dynamics is closer to the real life situation where both ligand and polymer structures are kept flexible. This mimics how the ligand and polymer exist in the body. Molecular dynamics studies provide a more precise understanding of the ligand-polymer interactions. In this case, the structure of both the polymer and curcumin were prepared using the Schrödinger-Mastro software. Although they could have retrieved the structure of curcumin from PubChem, they opted to draw it manually, which is perfectly fine. The structure of Udrugit, being a polymer, is not available on the protein data bank since it is not a protein. Remember that for formulation development studies, polymers replace proteins and drug molecules act as ligands. Docking was performed using the glide module of Schrödinger and the results are provided in the table. As we know that curcumin can exist in two forms, diketo or ketoenol, and Udrugit may be present as either a monomer or a dimer. Therefore, considering these possibilities, in this study, the docking was conducted using all these combinations. The glide G score is the docking score in this case, and it is always in negative. More negative score means higher the affinity between drug and polymer. In this case, it is lowest, that is minus 1.958 for the diketo form of curcumin with the Udrugit dimer. Now, what is this glide E model value? This value denotes the affinity and stability of the complex. In this case, higher the value, more is the stability. And we see that ketoenol form of curcumin with Udrugit dimer is having best stability out of all the combinations. The value of this score is minus 19.483. This confirms that Udrugit is forming complex in the form of dimer with both forms of curcumin. Now, if we look at the results of molecular dynamic simulations, they revealed that the van der Waals interactions contributed more to the total energy of complex than hydrogen bonds. This is consistent with expectations for solid dispersions. In solid dispersions, It is preferable for the drug-polymer interaction to be strong enough to enhance the solubility of the complex, yet weak enough to dissociate easily in physiological conditions. In conclusion, they noted that the in silico studies were consistent with FTIR spectra, and thus they used these studies to confirm their findings on drug-polymer interactions. In silico studies are relatively easy to perform and when used correctly can yield reliable results. This approach can be applied in formulation development whenever molecular interactions occur between two substances, such as drug polymer, drug excipient, drug stabilizer, or drug taste masking agents. While this study used in silico methods for confirmation, these studies can also be confidently employed to predict interactions between different species. To summarize, molecular docking can be utilized to study drug polymer or drug complexing agent interactions during formulation development. If multiple polymers are involved, it allows for the prediction of interaction strength in advance. In stability enhancement studies, which are aimed at preventing degradation of the drug molecule by protecting its vulnerable sites with the help of some protecting groups, molecular docking can identify which stabilizing agents interact with the drug in a manner that protects these critical areas. A similar approach can be employed to assess the compatibility of excipients with the drug. Ideally, the drug and excipient should not interact, and which should be evident in the docking studies. And when we are using docking for taste masking, it is important to ensure that the chosen taste masking agent interacts with the drug in such a way that the bitter tasting portions are shielded. Thus, molecular docking can help accelerate certain formulation development studies while providing a deeper understanding of the interactions between the drug and other essential components of the formulation. I hope that you found this video helpful in designing your studies related to formulation development. If you have any questions, please write them in the comment box. I will be very happy to answer them. If you are interested to know more about any topic related to research in pharmaceutical sciences, please share in the comment box. We shall meet soon for the discussion of another useful topic. Till then, keep learning, keep growing.